And we'll give folks just a moment to cycle in here before we get started. While everyone kind of filters in, I'll go ahead and get some introductions going. Hi, everyone. Good morning, good afternoon, or good evening, wherever you might be. If you're in the US, happy Election Day for those who celebrate. Welcome to the 7th week in the fall 2021 iteration of the long-term animal research seminar series. Thanks for coming. This week, we're going to hear from Dr. Erin Siracusa and Dr. Alycia Lackey. And we're going to hear from Erin first, but before I introduce her, I'll make a couple of quick announcements. If you are participating via Zoom, you should see a Q&A tab on the bottom of your screen. If you open that tab, you're going to be able to type any questions that you have, as well as see and vote on other people's questions at the end of the talk. We're going to, each talk, we'll go through those questions, starting with those that have the most votes. And then second, we're going to be posting recordings of these talks to YouTube, as always. So if you want to watch them again, or if you know someone who can't watch them live, you'll find them there. So I'm very excited to introduce our first speaker today, Dr. Erin Siracusa, a postdoctoral research associate at the University of Exeter in the UK. Erin received bachelor's degrees in biology and English from St. Lawrence University, where she worked with Erica Barthelmess, studying the impact of porcupines on the floral composition of forests of New York. She then received her PhD from the University of Guelph, where she worked with Andrew McAdam, studying determinants of fitness in the Kluane red squirrels. And today we'll hear a bit about a paper that Erin published in Current Biology this year about the impacts of social partners on squirrels' fitness, which is a really cool paper for which she received the award for Best Student Paper from the University of Guelph College of Biology Sciences, and I believe was also at least in part the topic of her Alycia Award-winning talk at ABS in 2018. So really cool paper, excited to hear about it. Erin currently studies the causes and consequences of age-based changes in sociality among the rhesus macaques of Cayo Santiago. And we'll hear about some of that today too. So I'm excited to welcome Erin, and I'll turn things over to her now.
Thanks so much, Matthew. Let me just get my screen shared here. Great. Well, again, thanks, Matthew, so much for that lovely introduction and for the invitation to speak as part of this seminar series. So as you can all probably tell from this introductory slide, my current research is really interested in this intersection between social behavior and aging. So both in how social behavior affects senescence across various systems and tissues, but also how the aging process, whether that's senescence or the positive effects of age, such as increased skills and experience, can affect how social behavior changes across the lifespan. But before I tell you about some of my recent findings using a really exciting long-term study of rhesus macaques, I want to first step back and tell you briefly about how I got interested in these questions in the first place. And that involves introducing you to a different long-term system, which I had the pleasure of working on as a PhD student, the Kluane Red Squirrel Project. So this work was supervised by Dr. Andrew McAdam and was recently published in Current Biology, as Matthew mentioned. So you can find the full story there if you're interested in learning more. But the upshot of this research was that we used this amazing long-term study on North American red squirrels, which are a highly territorial solitary species, where we had a data set spanning 22 years and over 1,000 individuals to show that social familiarity with territory neighbors increased fitness in red squirrels. So what you're seeing here in these figures is that red squirrels who had more familiar neighbors, so that is neighbors that they had lived next to for a long time, had a higher probability of annual survival and a higher reproductive success. So that's an increase in the number of pups sired for males and an increase in the number of pups recruited for females. And that simply means the number of pups that survived their first winter. So while these findings had really exciting implications for thinking about how long-term social relationships can affect even species we typically think of as being asocial, what was perhaps even more surprising to us was how important these familiar neighbors were in later life, or what we would consider to be the senescent period for red squirrels. So in these senescent models, the effective familiarity was basically on par with the magnitude of that of age in the same models. And this suggested that familiarity might help to offset the negative effects of aging. So another way you can think about this is if a squirrel were to age from 4 years old to 5 years old, our models suggested that its survival would decrease by 10%. But if that same squirrel were to age by a year, but maintain all of its neighbors so that its familiarity actually increased by a year, survival would actually increase by 6%. And so this suggests that familiarity, that is having these long-term relationships with neighbors, can help to buffer squirrels from these age-related declines in fitness. And so it's this finding that got me really thinking about what role these sorts of social relationships might play in the aging process. And this seems a particularly pertinent question, given that there's increasing interest in the substantial variation and patterns of age-related changes in survival and reproduction that we observe in the wild, and in trying to understand why we observe this variation and the processes underlying these declines. And so, as a result, there have been a growing number of studies that test for age-dependent decreases in other traits, including physical traits, immunological traits, molecular traits. But far fewer studies have focused on age-related changes in behavior, and in particular, social behavior, a process that's been referred to in the literature as social aging. Now, this lack of research on social aging is particularly striking, given that we know that social relationships are really central to the lives of group living animals and can have really substantial effects on health, survival, and reproductive success across a wide range of taxa. So as a result, understanding how social behavior changes with age is important, as it can modulate how fitness prospects change across the lifespan, and may therefore be an important source of unexplained variation in patterns of senescence. And so this is where my current research comes in. So I'm using a long-term population of rhesus macaques on the island of Cayo Santiago to understand this intersection between sociality and aging. And this current work is being done in collaboration with Dr. Lauren Brent here at the University of Exeter, Dr. James Hyam at New York University, and Dr. Noah Snyder-Mackler at Arizona State University. So today, I'll tell you a bit of a story about this sort of side of the arrow, looking at changes in social behavior across the lifespan and how we've been able to use this amazing long-term system to investigate one potential cause of these age-based changes. So one of the most common patterns of social aging observed both in humans, but also other animals, is the narrowing of social networks with age. So given this established importance of having strong social connections for health and fitness, this shrinkage in networks with age has typically been thought to be maladaptive and has been sort of ascribed to age-related losses. So things like poor health, deaths of social partners, or loss of social motivation. But more recently, it's been suggested that this narrowing of social networks with age might be driven by proactive processes. So the general idea in humans is that this sort of increasing awareness of limited time left in life leads individuals to sort of selectively prune their social networks and focus in on emotionally meaningful relationships like close friends and relatives. But what's particularly interesting is that recent research has also begun to demonstrate evidence of social selectivity in non-human primates that don't share this awareness of limited time. So this seems to suggest that there are sort of deeper evolutionary roots to these patterns of social selectivity, and that this narrowing of social networks with age might actually have adaptive value. But arguably, I would say it's been really difficult to conclusively show evidence of social selectivity in non-human animals. And that's because I would say there are sort of several pieces that need to be brought together to demonstrate this phenomenon. First, and perhaps most obviously, that social networks do actually narrow with age. Secondly, that narrowing is driven by the aging individual itself and not by others withdrawing from interaction with that older individual. Um, Third, that the aging individual continues to show engagement and interest in the social world to help disentangle social selectivity from a narrowing of networks that might result from declines in social interest or motivation, or even sort of a physical ability to engage. And fourth, that there's an increase in focus on important relationships with age. So in the biogerontology literature, important relationships have typically been viewed as family and close friends, since those are sort of the most emotionally salient relationships. But increasing focus on kin and strongly connected partners with age might also make sense from an evolutionary perspective. And finally, and perhaps most importantly, one of the major gaps in our ability to demonstrate social selectivity is that it is fundamentally a within individual phenomenon. If you can imagine that each of these different colors is a different individual, what we need to demonstrate social selectivity is that there is a within individual change in behavior across the lifetime. And most studies attempting to look at this to date have been cross-sectional in nature, meaning they observe different individuals in different age classes. And this makes it hard to know whether those purported age-related changes are actually due to age per se, or are the result of differences between cohorts or processes like selective disappearance. So this is where we've been able to leverage this really amazing long-term system where we have longitudinal data to better address these questions. So Cayo Santiago is an island off the coast of Puerto Rico, and it consists of approximately 1,500 free-living rhesus macaques living in about 6 to 9 mixed-sex social groups. So the monkeys are food supplemented, and they have no natural predators on the island. For this study, we used 8 years of data on female macaques 10 years and older. And so during this time, we collected 2 different types of social association data. So we collected grooming data using 10-minute focal animal samples. And we also recorded time spent in proximity by conducting 3 scans at evenly spaced intervals throughout that focal animal sample. So in total, this gave us 563 monkey years of data over 204 unique females. So on average, we had 2.8 years of data per female, but that ranged between 1 to 8 years per individual. So statistically, we used a within individual centering approach within a mixed effects model framework to look at changes in social behavior across an individual's lifetime. So I won't discuss that sort of methodology further here for the sake of time, but I'm happy to answer questions about it later. But the biggest thing to take away from this is that all of the results I'm about to show you are within individual changes. And finally, the last thing I want to mention about these models before I show you the results is that female rhesus macaques have a strict dominance hierarchy with maternal rank inheritance. So to account for the possibility that rank might affect these age-related changes in behavior, we also tested for a rank by age interaction, and we kept that interaction term in the models where significant. So what did we actually find? So to assess this first prediction that social networks do narrow with age, we looked at changes in the number of grooming partners and changes in the number of individuals that females spent time in proximity to with age. And in both cases, we saw significant declines with age as we predicted. So when we looked at change in the number of proximity partners, we did find that this effect was rank dependent. So lower ranking females decreased their number of proximity partners more strongly with age than did high ranking females. And just to orient each of these figures and those that will come after, so here the points represent the raw data. The solid black line shows the average predicted within individual change with age, and these differently colored slopes show the inter-individual variation in this pattern. So all of these models included a random slope term, and those differently colored slopes are basically the visual representation of that random slope. So to assess the second prediction, to show that this reduction in number of partners is driven by the aging individual, we looked to see if the number of partners approached by the aging individual declined with age. And we found that it did. Again, this relationship was rank dependent, so lower ranking individuals reduced the number of partners they approached more strongly than did high ranking individuals. We complemented this by then looking to see if the number of partners that an individual was approached by remained consistent with age, and we found that it did. So what this seems to suggest to us is that while females actively approach fewer individuals as they get older, they continue to be approached by similar numbers of partners, suggesting that this narrowing of networks is not being driven by the social withdrawal of others. Regarding the third prediction, so continued engagement in the social world, we expected that even though individuals were narrowing the number of partners they interacted with, the amount of time spent giving and receiving grooming would remain constant with age. And in both cases, we found that it did. So there were no changes in grooming given or grooming received as individuals got older. Similarly, we look for changes in time spent in proximity with age, and again found that it remained consistent as we predicted. So this collectively seems to suggest that even though individuals are reducing the number of partners, they still remain interested and actively engaged in the social world. And finally, to look at whether female macaques focused in on important relationships as they aged, we first assessed whether they prioritized kin with age. And we found that indeed they increased their proportion of kin partners as they got older. And this is even accounting for the fact that availability of kin does increase with age. Furthermore, we know from previous research that female macaques, both strong dyadic connections and connections to stable partners are significant predictors of survival. So that suggests that perhaps both strong and stable partners are important relationships to maintain. And so in line with this, we found that individuals who were on average more strongly connected to the focal individual earlier in life were more likely to be a partner in later life. Similarly, we expected that females would narrow their networks to their most stable partners. So here, a stable partner is any individual who a female was partners with for at least 2 consecutive years. And again, this is what we found. So females were more likely to choose partners with whom they had previously had a stable social relationship with earlier in life when choosing who to focus their social effort on in later life. So to conclude, we've found evidence for all of these predictions, suggesting that female rhesus macaques do show evidence of social selectivity with age. Now, arguably, I would say this is the most comprehensive test of social selectivity to date in a natural population, and one of the few studies to show that these patterns result from within individual changes in behavior. So this adds to a growing body of evidence showing that reductions in number of partners with age is not exclusively the consequence of declines in bodily systems, but that older individuals might actively alter their social choices to cope with new challenges or take advantage of new opportunities in later life. So understanding how and why social behavior changes with age and further investigation of whether those changes have fitness benefits or costs will offer important insight into how social behavior might then be expected to feed back and affect other patterns of senescence. And so finally, these results also suggest that these patterns that we observe in humans may not simply result from these sort of complex cognitive mechanisms, such as awareness of limited time, but might indeed have sort of deeper evolutionary underpinnings. So that's it for today. Thanks so much for listening. If you're interested in learning more about how I use long-term mammal systems to investigate the effects of social interactions on behavior, fitness, and patterns of aging, you can head over to my website or follow me on Twitter. A really big thanks to our funding and especially to all of the research assistants and the CPRC staff who've been instrumental in our efforts to collect these data over the past 10 years. So thanks again, and happy to take questions if we have time.
Great, thanks very much. That's a very fascinating talk. I have a question. I'm going to defer to the Q&A for now, and I'm going to ask mine later. So Emily Levy asks, she says, wonderful talk, thank you. In your grooming given plot, prediction 3, I think, it looked like there was a lot of inter-individual variation in grooming given early in life and then less later on, less variation later on. Do you think anything interesting is going on there?
Yeah, so I think there's a lot, I think, to explore in terms of these patterns of inter-individual variation in aging. And to speak sort of broadly to your question, Emily, I think this is sort of the next step in some of these analyses, is to think about both what might be some of the causes of this inter-individual variation, but also what are some of the consequences. So it would be really interesting to see whether these changes in physical abilities or energetics or sensory systems correlate with these social changes, and similarly, whether sort of different patterns of social aging have different fitness consequences. And I think the rhesus macaques offer a particularly nice holistic system to investigate this. the project that I'm currently involved in on CAIO is collecting not only behavioral data, but longitudinal, morphological, physiological, genomic data to see how these changes across various systems correlate with social changes, and importantly, whether they sort of precede or follow those social changes. So I think more to come on that front. But the other thing I should note is that these random slopes actually in all of these models explain only a very small portion of the variation in the model. So in all cases, really less than 1%. So despite maybe perhaps appearances that there might be a lot of inter-individual variability in these patterns, it might not be the case. So yeah, stay tuned.
And then I'm going to ask you to walk way out into speculation land. You indicated a couple of times that you're assuming that non-human primates are not aware of a shortening of the amount of time they have left to live. How confident are you that that's true?
Well, I'll be honest, Matthew, I'm pretty new to the primate world, as you might have sort of seen from my previous research. And so, I can't profess to be super well versed in that literature. But it seems to be a pretty well-established sort of theme in the literature that we don't think that primates have this awareness of, non-human primates have this awareness of limited time. So I would venture to say that I think there are probably other drivers of this social selectivity that don't necessarily involve that, but hard to say for sure.
Fair enough. Well, thanks for that. I think probably almost everyone does assume that, right? I'd be eager to see if there's an empirical way to get at that.
Yeah.
Anyways, thanks very much. A really great talk. Thanks for coming and giving it. Our next speaker today is Dr. Alycia Lackey, assistant professor in the Department of Biology at the University of Louisville. And in preparing for today, I was terribly excited to learn that Alycia and I share the biology department at UNC Chapel Hill as our undergraduate alma mater. And beyond that, we share Corinne Fenig as our undergraduate research advisor. So that was a really fun. There's always these kind of small world effects in academia, and that was a cool thing to learn. She received her PhD from Michigan State University, where she worked with Jenny Bauman. And she then held postdoctoral positions at Murray State and Binghamton University before joining the faculty at Louisville last year. Alycia and her lab do a really admirable job of integrating lab and field work to measure phenotypes, selection, and fitness in wild populations. And today we're going to hear about a really exciting long-term study of tiger salamanders. That's right, after 53 talks in this series, we're finally going to host a talk about amphibians. I'm really excited about that. So I think Alicia's work really captures the breadth of the research that we hope to display in this series. And I'm so glad that she's able to join us today. And I'll hand things over to her now.
All right. Well, thanks so much, Matthew. That was a really nice introduction. And it is so nice to be part of this seminar series and to get connected with other people thinking about these questions in long-term data sets. And so it'll be interesting also to think about how these kinds of questions in amphibians might overlap with things that are getting asked in other systems as well. So today I'm going to tell you about work from one of my postdocs looking at lifetime fitness and life history strategies in these tiger salamanders. And while I did this work as a postdoc, this is also ongoing work. So I'm still collaborating with this research team to analyze the data and ask new questions. And we're also working on some collaborative funding proposals, especially extending this work that I've done about lifetime fitness consequences and looking at this in a climate change context, because as we have these long-term data sets, it's a really nice opportunity to be able to ask how environmental changes over long periods of time can influence the patterns that we see in these systems. I also wanted to set the stage with the kinds of questions that my research and my lab has been interested in. And so in the big picture, we ask about how the environment affects multiple different levels across the organismal level, thinking about physiology and behavior and plasticity at the level of evolution, how genetic changes can help explain patterns in reproduction and life cycle timing, and other variables. And then at the ecological level, thinking about competition and species interactions and habitat use. And then, of course, lots of interactions between all of these levels. And the big picture kinds of questions that I ask in my lab are responses to environmental change, like populations' capacities to adapt, mechanisms, ecological and evolutionary, to respond, and also bringing in the community levels and feedbacks between the population and community levels. And then from more of an evolutionary perspective, I ask questions about how differences within and between populations evolve and what kind of consequences that has for the evolution of differences within and between populations, but also maintenance of variation and consequences for biodiversity. So today's emphasis in the tiger salamanders focuses on this question, that is, how can variation evolve and persist within populations? I've also worked on other systems that think more about the evolution of distinct species and how that process happens. But the tiger salamanders give us a nice window to think about how variation can evolve and be maintained within populations. So now the diagram shows sort of a subset of, in each of these categories, the kinds of things that I'll be focused on. So this study system and this question focuses on phenotypic plasticity and the evolution of reproductive life cycle timing and plasticity, and then how competition within species influences all those dynamics. And here, environmental change is more of, rather than the climate change perspective, we're really focused on natural fluctuations in these populations over time and how some of those dynamics influence responses to environmental change at these different levels. And then the big picture evolutionary questions are thinking about how we get variation persisting and evolving within populations. So in this tiger salamander system, there are two morphs that are produced as a plastic response to variation in the environment. And phenotypic plasticity occurs when we have a single genotype that can produce different phenotypes in response to different environments. So like in this case of the predator-induced defenses in Daphnia. And we would expect plasticity to be favored when an environment, when a plastic response to the environment improves fitness of the organism more than a fixed phenotype given your genotype. And we can see plasticity as a continuous phenotypic response across different environments. So for example, as temperature increases, we could see earlier days of flowering. But you can also get discrete plastic responses. So here, phenotype A versus phenotype B, where you can have two or more distinct phenotypes that are produced across environments. And that kind of plasticity is called a polyphenism, where you produce these distinct morphs. And so here are a couple striking examples of diversity within populations, including spadefoot toad larvae that have two feeding morphs, short and long-term mating morphs in dung beetles, and these peppered moth caterpillars with a color polyphenism. And often we find that these different morphs are associated with alternative life histories, like time to development or size at maturity, because there are environment-specific payoffs of these different strategies. So these polyphenisms give us an opportunity to ask, how fitness consequences, or what are the fitness consequences for each morph? We can gain a better understanding for how selection might maintain phenotypic plasticity within a population. And it allows us to explore how plasticity produces diversity. And so also think about the potential for plasticity to promote divergence within and between populations. So in the tiger salamander system, larvae can develop into one of two morphs. They can develop into a pedomorph, which retains the larval characteristics like its gills, and it stays in its natal pond where it was born for its entire lifetime. It can also develop into a metamorph that goes through metamorphosis like many amphibians. So they leave their natal pond and feed and overwinter in the surrounding terrestrial environments. And then in the summer, they return to these ponds to breed. And we find with equal likelihood that these morphs will breed with each other. So there doesn't seem to be a mating limitation in terms of morph interactions. And from previous work, we know that different levels of competitor density experienced by these larvae is what triggers the production of one morph over the other. So in general, when competitor density is low, then it's beneficial to stay in the pond and develop as a pedomorph to take advantage of these plentiful resources. But when competition is high, salamanders benefit from escaping the ponds as metamorphs. And we also know that this competitor density varies across time and space, which I'll talk about at the end of the talk today. So my main focus today is to think about how differences in fitness and life history might occur between these two morphs and how that helps us potentially answer this question about what maintains this polyphenism. So first, I measured lifetime reproduction of each morph to think about the adaptive significance of this polyphenism. So are there fitness benefits to each morph that maintain them in the population? I also measured differences in the life history strategies of these morphs, looking at traits like development time and longevity to evaluate which strategies predict high fitness in each morph. And then lastly, I examined the idea that environmental variation could maintain this polyphenism if the fitness of each morph differs across space or time. And this work happens in a population of Western tiger salamanders that is isolated at high elevation in the Rocky Mountains in Colorado, near the Rocky Mountain Biological Lab. And most of the year, the area is covered in snow and ice, and there's a 3-month growing period and breeding period in the summer. And it is an absolutely beautiful field site. So I'm always looking for more excuses to go back. This is showing an array of different ponds at the site that vary in their size. So here's a particularly big pond and one of the smaller ponds, for example. They also differ in their permanence. So whether they stay, there's water all year round or whether they dry up from year to year. So there's variation across space in these habitats that are available to the salamanders. And this is one of the largest ponds at the site with one of our team members going to collect salamanders. And all of the ponds are very shallow. So we go out and collect primarily by netting the salamanders or go out in small boats and net them from there. So this long-term monitoring project has been going on since 1988. So we have over 30 years of data now. And there's lots of people to thank who have contributed to that project, especially Howard Whiteman, who's been leading this effort for decades, and a lot of his collaborators, his undergraduate and master's students, postdocs and faculty. And we have lots of funding sources to thank as well. So most of the larvae and adults in this population are individually marked with PIT tags. So they have these tags under their skin that we can scan and identify them by a serial number and enter their information into the database each time they're caught. And what I did was take 27 years of data, which encompassed over 32,000 captures of these salamanders in that time period, and analyzed individuals where we knew what year they were born in. So they have a known cohort, and that resulted in data from over 6,000 individuals. And then in this, the general collection approach, every time an individual is caught, we measure a series of life history traits across their entire lifespan. So we measure their body size, their reproductive maturity, what morph they are, whether they're in reproductive condition at that time. So these are these captures are done across the summer in every year. So you can also track potentially differences within a year. And for fun, this is one of the biggest salamanders in the data set. She has since passed on, but this female, when she was 25 years old, weighed 100 grams. So she's a really sizable, big salamander. And so that, like that salamander who lived 25 years, some salamanders can live quite a long time, but they don't all live that long. So the larval stage is typically about 3 years. So there is this long period to maturity. And then adults can live as long as about 30 years, but as we keep monitoring this population, we keep having to increase that, that total lifespan. So today I focused on analyzing our oldest cohorts where we have the most data for them. So salamanders that were born between 1988 and 2001 with 14 to 27 years of data. And so that encompasses most of their lifetime data on their growth, survival, and reproduction. And to start with the lifetime fitness measures, I looked at that separately for the sexes because biologically males are going to be they're going to have the advantage by increasing their reproductive success through multiple matings, whereas females increase their reproductive success by making more eggs. So here's what things look like for males. We have the two morphs, paedomorph and metamorph on the x-axis, and then the male reproductive opportunities on the y-axis. So this is every year that they were in reproductive condition and could have had a breeding event. So these are opportunities for breeding. And you can see there's tons of variation from 0 experienced by many individuals all the way up through 10 years of potentially breeding for some individuals. And there's also a significant morph difference. So if we zoom into just the means with their 95% confidence intervals, even though the confidence intervals overlap, we have such large sample sizes that we can detect a difference between these means. And what this equates to, if you think about the number of offspring that a male could sire in each reproductive event, on average, paedomorphic males could have 35 more offspring in their lifetime. So even though this difference in the number of reproductive opportunities might seem small, it could have a sizable fitness advantage. And then for females, we looked at estimated number of eggs she could produce in her lifetime. And we did that by taking size measures and actual egg numbers for a subset of 80 females from this population, and then use the regression line to predict for all the other females in the data set that based on her body size, how many eggs would she be predicted to produce in each year that she's in reproductive condition. And so that's what these data are. Now we have each of the morphs with female lifetime egg production estimated on the y-axis. So tons of variation again, lots of individuals at 0, many around the 200 to 400, and then some individuals having close to 1,500 potential offspring in their lifetime. And so what we see here is now there's a metamorphic advantage. So they're producing more eggs per lifetime on average than paedomorphs, with about a difference of 133 more offspring per female. And so to summarize this piece, we have sex-specific fitness advantages that differ between the morphs. So paedomorphic males have more reproductive opportunities, while metamorphic females produce more eggs per lifetime. And so these estimates are proxies for their lifetime reproductive fitness. And we would need maternity-paternity analysis and actual numbers of surviving offspring to even better measure these fitness advantages. But it still provides some insight that there are sex differences between the morphs. That could be one mechanism that maintains this polyphenism. So next we asked about how life history strategies between the morphs could differ. And so we asked about multiple fitness components: longevity, age at first reproduction, adult condition, larval condition, and how each of those both connect to each other and predict lifetime reproduction. And so we used complementary approaches of multiple regression and path analysis. And so I'll show you what the path analysis looks like. So this is a hypothesized diagram of the relationships among these variables. And so we can test how number of reproductive opportunities, which is our measure of lifetime reproduction, is predicted by these other life history traits. And path analysis is based on linear regression, but it allows you to analyze both direct effects and indirect effects of the variables. So for example, we can look at the direct effect of age at first reproduction on number of reproductive opportunities. But we can also look at its indirect effects through longevity. So that's one of the benefits of path analysis, especially if you have lots of interrelated variables or nested variables. And what I'm showing you here is the best fit model. So we did test a number of different hypotheses that has the presence or absence of some of these paths here. But this one is the one that's best supported by the data. So we looked at a larval condition, which was measured in the year before they mature into adults. We measured age at first reproduction as their first year cotton breeding condition. Adult condition that's averaged across their lifetime. Longevity as the measure, the difference between when they were born, their cohort, and when their known death year is, or their last capture. So that could be a potential underestimate of how long they live. And then number of reproductive opportunities for both sexes. Now we look at the number of years in reproductive condition. And we also found that the number of captures per adult does not differ between the morphs. So our ability to detect variation in these variables should be similar across both morphs. All right, so of that whole path diagram, I'm just going to focus on what's going on at the adult level. And so now I'm showing the relationships as they're supported by the data. So thicker arrows are stronger relationships between the data. And solid arrows are all positive relationships, and this negative, this dashed line is a negative relationship. So there are two main paths to getting to high numbers of reproductive opportunities. You can either breed earlier, so this is a negative relationship where a lower age at first reproduction predicts higher number of reproductive opportunities. Or you can have a longer lifespan. So the longer you live, the more reproductive opportunities you can have. And there's also this relationship between age at first reproduction and longevity. So when you breed earlier, you tend to have a shorter lifespan. But if you delay your age at first reproduction, you tend to have a longer lifespan. So this relationship also suggests that there's going to be a trade-off between these two paths, that you can't do both. You can't breed really early in your lifetime and have a really long lifespan. And what we find in the data is there's a morph difference in their age at first reproduction and in their longevity, such that the morphs are taking these different paths to maximizing reproductive opportunities. So pedomorphs breed earlier and die earlier, and metamorphs breed later and die later. So they have these different strategies. And here's what these data look like. So you can see there's a lot of overlap, that both morphs can breed early in their lifetimes and can have similar lifespans as well. But there's also this difference between the morphs, and that metamorphs seem to be able to take advantage of this phenotypic space out here, where they can delay their age at first reproduction and potentially live much longer than pedomorphs have ever been detected to do. And there's also proportionally a bigger piece of the metamorphic population, or the metamorphic group, that takes this sort of delayed strategy, compared to very few pedomorphic individuals that do that. All right, the last piece here in connecting these hypotheses to what could maintain a polyphenism is to think about effects of environment and how they might vary over space and time. So what I'm showing you here are the cohort years, the years in which the salamanders were born, and the number of individuals we see from each of those cohorts. And then over space, we have the ponds that they're born into and the number of individuals in each of them. So here the bars are showing the number of pedomorphs and the number of metamorphs for each cohort. And one of the big things you can see is that this population goes through these boom and bust cycles. And we tend to see that about every 10 years. And that is related to this big production of pedomorphic individuals eats most of the hatchlings and larvae from the next cohorts that come up after it. And you have to wait until a lot of these pedomorphs die off before you can get another round of individuals that so there's breeding in every one of these years and hatchlings produced, but they don't survive to adulthood. And another piece here is that the morphs frequency differs across these cohorts. So the relative proportion of metamorphs to pedomorphs differs across these years. So you can imagine how these different frequencies could play into frequency dependent fitness of these different life history strategies or of these sex specific fitness advantages. And then across space, across these different kinds of ponds, which again differ in how big they are, how permanent they are. They also differ in the prey quality and the prey composition. So we also see across space, across these different ponds, that there's variation in the frequency of morphs that are produced, and also in the total number of salamanders that are produced across those ponds. So to step back for a second and think about how environments can influence a polyphenism, we could find that two different morphs have equal lifetime fitness across different environments, and that could maintain a polyphenism because they're equally successful. We could also find that lifetime fitness differs across environments. So orange is better than blue in environment A, and blue is better than orange in environment B. And so you can have overall differences in the lifetime success of these different morphs. But all you really need to maintain the polyphenism is that one has an advantage in one environment over the other, and then the reverse in here. So if we think about these averaged across the environments, even though overall it looks like the blue might not be as successful, it is more successful than orange in environment B, for example. And so to come back to this path analysis, just to illustrate what some of the environmental effects are, I won't show all the details, but I'll just sort of highlight which environmental effects play out across these life history traits. So we found that there were no effects of cohort or of pond on number of reproductive opportunities overall, but that cohort and pond affect each of the other life history traits that play into explaining variation in number of reproductive opportunities. So these cohorts and ponds have a big impact on larval condition. What year you were born in affects your adult condition. The pond you were born in affects how early you breed in your life, and there are cohort and pond effects on longevity. So it's interesting to think about how these earlier life history traits are environmentally sensitive, and then how that predicts these number of reproductive opportunities. So to summarize, we looked at multiple aspects of what could maintain this polyphenism in the salamanders. We found alternative life history strategies between pedomorphs and metamorphs. We find these sex differences within each morph, and we also find impacts of environmental variation. And all those pieces playing together can contribute to maintaining this variation within a population. And I wanted to highlight how critical it is that we have this long-term data, because otherwise we wouldn't be able to see some of these environmental patterns over time, or look at life history or fitness differences in such a comprehensive way. So I'm always excited to hear about other people's long-term data because of the kinds of questions that opens up that we can ask. So thanks so much for listening.
Great, thank you very much. This is such a fascinating system. I'm trying to wrap my head around it a little bit. let me start with the question, and then Ron Grunwald has a question. I think he could just ask it live. I'll let him ask it live. So I'm just trying to kind of wrap my head around the idea that there's just no adults surviving for the middle 6 years. How does that tie into like the decision-making process for when to start reproducing for these animals? Are they kind of assessing the number of pedomorphs in the environment?
Yeah, so one thing that I can say to address that is that so pedomorphs aren't going to have much of a choice about breeding, right? They're in the pond that they're in, and so you either breed in that year or you don't. I don't know about that decision making, but metamorphs will often visit multiple ponds before reproducing in each year. And so they could be assessing, density in those ponds. They could be assessing prey quality. We don't know, but we do know that they can visit multiple ponds, and they tend to have preferences for reproducing in certain ponds over others. So there's a lot of avoidance of pedomorph heavy ponds. And we don't know how they're detecting that, but they tend to breed not in those ponds. So yeah, it is really interesting, and to think about that the morphs have different opportunities to have strategies to avoid being eaten, having their offspring eaten from year to year. So
I'm going to try something new here. I'm going to hit the allow to talk button for Ron Grunwald. I've never tried this, so let's see what happens. Ron, I think you have to unmute yourself, but
I think I unmuted myself. Did that work?
Oh, cool. Great.
Great. Okay. Thank you, Alycia. Thanks very much. Cool stuff. I think actually you just touched on my question, which had to do with dispersion, and particularly dispersion by males. And the first question, I just want to confirm that in sampling, you're not missing individuals because they're dispersing out of your sampling area. I assume you're sampling and aggregating from all of the ponds. I'm gesticulating with my hands, you can't see that, but from all of the ponds, and that there's not an opportunity for them. This is kind of a unique setting if it's the case that they don't have an opportunity to disperse beyond. And the other question is, do you see patterns of dispersion, and do patterns of dispersion by males have an influence on their reproductive fitness, particularly if they're outcrossing as a consequence?
Yeah, those are great questions, Ron. So to speak to the dispersal one, this series of ponds is really the only available habitat for these salamanders to breed in like many kilometers. So like down about 8,000 kilometers, there's another population of tiger salamanders that are down there, but there's no mixing between these. So by sampling across all the ponds each year, and because metamorphs, if they're going to breed that year, will have to come back to these ponds to breed, we do feel like we are sampling this whole population. Now, there can be years where metamorphs don't reproduce. They tend to take longer between each reproductive event. So even if we aren't detecting them that year, that doesn't mean that they aren't still alive and won't reproduce in the future. So
presumably it means they're not out having fun in another pond that you're not sampling.
Yeah, right, because we do sample across all the available ponds. Yeah, and then remind me of your second question.
Oh, just whether or not dispersion within the cluster of ponds that you are sampling, how do you account for impact on reproductive fitness related to inbreeding versus outbreeding, and the fact that the point here being that metamorphs can disperse and pedomorphs cannot?
Yeah. So this work connects more with a colleague of mine, Michael Moore, who's a postdoc now in St. Louis, and he has looked at pond fidelity. So whether, I think their patterns were primarily for males. We didn't seem to see a pattern for metamorphic females showing a preference for ponds, but I think that males were showing a preference for ponds that was also dependent on their their body condition as larvae. So there could be something where they're using that, there's information potentially available about how good your early life was and whether you would return to that pond to breed. So there is some work thinking about that and whether they're mating in their natal ponds or in other ponds. But metamorphs certainly do have that opportunity to assess pond quality while paedomorphs can't make those movements.
Right. I'm thinking of the work of alpine newts in Switzerland and where dispersion for outbreeding seems to be a driver for the paedomorph to metamorph transition for males.
Oh, yeah. Yeah.
You know that system, I assume.
Yeah, a little bit. So I don't know that we have data to, compare exactly to those questions. I think the study that I just mentioned is probably the closest thing we have to looking at, which ponds they return to and breed in. But it's a really great question.
Great, thanks so much.
Yeah.
Thanks for that, Ron. Let's see, we have another question. Let's see. From JJ Bryson, who says, I've been thinking about demographic shifts and variation in cooperative strategies, which also seem to be morphs in human populations. So the fact that an ecosystem is almost never actually at equilibrium and that strategies should vary depending on when there are more or fewer resources than you expect seems universal across kind of environments. So the paedos seem to be born into good times and so can be conservative and stay at home, whereas the metas seem to be born into harder times to go in with more varied reproductive strategies. Does that make sense? It's what I understand about C. elegans with their different long versus short life histories, the longer being related to stress, so therefore postponing fertility.
Yeah, those are really interesting connections. And I think that does make sense in this system that in years where there are lots of resources available, that's going to support big adult populations of these paedomorphs, whereas tighter years will either produce no adult individuals or individuals that have high enough condition to metamorphose will metamorphose out of those ponds in competitive environments. But one of the things I didn't talk about is that metamorphosis is size limited. And so if it is a really tough year, an individual that doesn't reach that size threshold to metamorphose basically just has to, develop for a whole nother year with the hope that in the next summer they might be big enough to metamorphose. So there are certain individuals that can't reach that size threshold and so just have to wait it out and see if they can survive another year before reproducing. So, yeah.
Great. Thanks for that question, Joanna, and welcome. I think that if there's no more questions, this has been really, this is a system I didn't know anything about, and I'm really excited to know about it. And I'm going to spend some time, I know, thinking about the way that kind of this, it's just so different the way that there's so much variation in lifetime success based on the year in which an animal is born. And that's just so different from the way that I think about things. And I'm not really sure what the question is, but I guess I'd just be interested to kind of close out hearing how, how it shapes your view of a species where the time and place where an individual chooses to reproduce has such a huge impact on their actual fitness success. Yeah,
and I think that this system was really eye-opening for me as well, because, as a population biologist, in my development during my PhD, I really thought about, each year that we collected data and what are we understanding about the population as a whole. But, anybody that's collected data for more than 1 year, you often see a lot of variation in the things that the variables that you're interested in. And so I try to keep that in my mindset now about what are we measuring here, but what is the other variation that could be going on? And during my PhD, when I was working with stickleback fish, I did some comparative work across different labs that had measured things in different years. And there are often different strengths of selection or different amounts of isolation between these two species that we were looking at, depending on the year or depending on the location. And so that was also starting to get me thinking about this these kinds of variation that we don't always get to capture because of, because of limitations in how we can go out and measure things in these systems. And so that's just another benefit of these long-term data sets, because we can potentially capture some of those dynamics of variation.
Great. Well, I think that that's a great place to leave things. Thanks to you both for these really fascinating talks. And I think they both really speak to the unique benefits of long-term systems. So thanks to you both for being here. And thanks to everyone out there for joining us. We're going to be here in the same time and place next Tuesday, and then for 2 more Tuesdays after that. So we'll hope to see you then. Thanks to you both.